Ultracentrifugation is a process that involves the use of the centrifugal force for the sedimentation of heterogeneous mixtures with a centrifuge, used in industry and in laboratory settings. This process is used to separate two immiscible liquids. More dense components of the mixture migrate away from the axis of the centrifuge, while less dense components of the mixture migrate towards the axis. Chemists and biologists may increase the effective gravitational force on a test tube so as to more rapidly and completely cause the precipitate to gather on the bottom of the tube. The remaining solution is properly called the supernate, or supernatant liquid. The supernatant liquid is then either quickly decanted from the tube without disturbing the precipitate, or withdrawn with a Pasteur pipette. The rate of centrifugation is specified by the angular velocity measured in revolutions per minute, or acceleration expressed as g. The conversion factor between rpm and g depends on the radius of the sample in the centrifuge rotor. The particles' settling velocity in centrifugation is a function of their size and shape, centrifugal acceleration, the volume fraction of solids present, the density difference between the particle and the liquid, and the viscosity. The most common application is the separation of solid from highly concentrated suspensions, which is used in the treatment of sewage sludges for dewatering where less consistent sediment is produced. In the chemical and food industries, special centrifuges can process a continuous stream of particle-laden liquid. Centrifugation is the most common method used for uranium enrichment, relying on the slight mass difference between atoms of U-238 and U-235 in uranium hexafluoride gas. Centrifugation in biological research, microcentrifuges. Microcentrifuges are used to process small volumes of biological molecules, cells, or nuclei. Microcentrifuge tubes generally hold 0.5 to 2.0 milliliters of liquid, and are spun at maximum angular speeds of 12,000 to 13,000 rpms. Microcentrifuges are small enough to fit on a tabletop and have rotors that can quickly change speeds. They may or may not have a refrigeration function. High-speed centrifuges, high-speed or super-speed centrifuges can handle larger sample volumes, from a few tens of milliliters to several liters. Additionally, larger centrifuges can also reach higher angular velocities. The rotors may come with different adapters to hold various sizes of test tubes, bottles, or micro-titter plates. Fractionation process, general method of fractionation, cell sample is stored in a suspension which is buffered, neutral pH, preventing damage to the structure of proteins including enzymes, isotonic. This prevents water gain or loss by the organelles, cool, reducing the overall activity of enzyme released later in the procedure. Cells are homogenized in a blender and filtered to remove debris. The homogenized sample is placed in an ultracentrifuge and spun in low speed. Nuclei settle out, forming a pellet. The supernatant is spun at a higher speed. Chloroplasts settle out. The supernatant is spun at a higher speed still. Mitochondria and lysosomes settle out. The supernatant is spun at an even higher speed. Ribosomes, membranes settle out. The ribosomes, membranes and Golgi complexes can be separated by another technique called density gradient centrifugation. Ultracentrifuges. Ultracentrifugation makes use of high centrifugal force for studying properties of biological particles. Compared to microcentrifuges or high-speed centrifuges, ultracentrifuges can isolate much smaller particles, including ribosomes, proteins, and viruses. Ultracentrifuges can also be used in the study of membrane fractionation. This occurs because ultracentrifuges can reach maximum angular velocities in excess of 70,000 rpms. Additionally, while microcentrifuges and supercentrifuges separate particles in batches, ultracentrifuges can separate molecules in batch or continuous flow systems. In addition to purification, analytical ultracentrifugation can be used for determination of the properties of macromolecules such as shape, mass, composition, and conformation. Samples are centrifuged with a high-density solution such as sucrose, cesium chloride, or iodoxanol. The high-density solution may be at a uniform concentration throughout the test tube or a varying concentration. Molecular properties can be modeled through sedimentation velocity analysis or sedimentation equilibrium analysis. During the run, the particle or molecules will migrate through the test tube at different speeds depending on their physical properties and the properties of the solution, and eventually form a pellet at the bottom of the tube, or bands at various heights. Other applications: separating chalk powder from water, removing fat from milk to produce skimmed milk, separating textiles, removing water from lettuce after washing it in a salad spinner, separating particles from an airflow using cyclonic separation, the clarification and stabilization of wine, separation of water particles from clothes while spin drying in washing machines, separation of urine components and blood components in forensic and research laboratory. History: by 1923 Theodor Svedberg and his student H. Rind had successfully analyzed large-grain sols in terms of their gravitational sedimentation. Sols consist of a substance evenly distributed in another substance, also known as a colloid. However, smaller grain sols, such as those containing gold, could not be analyzed. To investigate this problem, Svedberg developed an analytical centrifuge, equipped with a photographic absorption system, which would exert a much greater centrifugal effect. In addition, he developed the theory necessary to measure molecular weight. During this time, Svedberg a unregistered trademark's attention shifted from gold to proteins. By 1900, it was generally accepted that proteins were composed of amino acids. However, whether proteins were colloids or macromolecules was still under debate. One protein being investigated at the time was hemoglobin. It was determined to have 712 carbon, 1,130 hydrogen, 243 oxygen, 2 sulfur atoms, and at least 1 iron atom. This gave hemoglobin a resulting weight of approximately 16,000 R, but it was uncertain whether this value was a multiple of 1 or 4. Through a series of experiments utilizing the sedimentation equilibrium technique, 2 important observations were made. Hemoglobin has a molecular weight of 68,000 R, suggesting that there are 4 iron atoms present rather than 1, and that no matter where the hemoglobin was isolated from, it had exactly the same molecular weight. How something of such a large molecular mass would be consistently found, regardless of where it was sampled from in the body, was unprecedented and favored the idea that proteins are macromolecules rather than colloids. In order to investigate this phenomenon, a centrifuge with even higher speeds was needed, and thus the ultracentrifuge was created to apply the theory of sedimentation diffusion. The same molecular mass was determined, and the presence of a spreading boundaries suggested that it was a single compact particle. Further application of centrifugation showed that under different conditions the large homogeneous particles could be broken down into discrete subunits. The development of centrifugation was a great advance in experimental protein science. Sources. References.